Hello listeners, Amynah here. Welcome to the Dr. GPCR podcast. In this episode, I talked to Dr. Amynah Pradhan from the University of Illinois at Chicago. Amynah's training started at McGill University, where she completed her PhD. Her next career steps took her for a postdoc in industry, one in Dr. Brigitte Kieffer's lab, and finally to UCLA, where she worked with Dr. Chris Evans. That is where her collaboration with Dr. Andrew Charles led her to decide to specialize in animal models of migraine and delta opioid receptors as therapeutic targets to treat headache. Before we jump into this episode, I wanted to take a moment to thank you for attending the first Dr. GPCR Summit. With 30 talks and 400 registrations, we are thrilled to say that it was a great success, upon which we are already building our next year's Dr. GPCR Summit 2021. You will shortly receive a survey from us in which we kindly ask you to tell us more about your experience at the summit and help us bring you a great summit in September 2021. And now, let's dive into our episode with Dr. Amynah Pradhan. Hello, everyone, and welcome to the Dr. GPCR podcast. In this very episode, I'm happy to have Dr. Amynah Pradhan as my guest. Hi, Amynah. How are you?
I'm good. Thank you. Nice to be here.
Thanks for being here today. So you're an Associate Professor of Psychiatry at the University of Illinois in Chicago. Can you tell us a little bit more about how you got to where you are today?
Yeah, sure. So basically, I started at McGill University, and I did an undergrad research summer internship where I was studying I was measuring inositol phosphates in airway smooth muscle cells. So it was really like biochemistry, lots of columns and things like that. And then I went on to do my PhD, like at McGill in the Department of Pharmacology and Therapeutics with Paul Clarke. And there, although he was a nicotine guy, I ended up doing my PhD on opioid receptors and the interaction between mu and delta opioid receptors. After that, I took a little bit of a detour and I did a one-year postdoc at AstraZeneca where I studied a lot of different animal models and endpoints in those animal models of pain, along with an SNSR G protein coupled receptor and its role in inflammatory pain mechanisms. And then I went back to opioids and I did a postdoc with Brigitte Kieffer in Strasbourg, which was great. And that's really where I got into ligand-directed signaling at the delta opioid receptor and its in vivo consequences. And then I followed that up with another postdoc with Chris Evans at UCLA, where we expanded that work looking at the role of arrestins in that ligand-directed signaling of the delta opioid receptor. And actually, that was a really important place for me because not only did it allow me to really broaden that work out, but I also started collaborating with a headache physician scientist, Andrew Charles, and he really got me interested in headache and migraine and looking at the role of opioid receptors within this totally novel pathophysiological state for me.
Wow. So you've traveled around the world
Yeah,
following your passion to study GPCR receptors and opioid receptors.
Yeah.
That is awesome. So you're studying right now opioid receptors in the context of headache and migraines. You had mentioned that you worked on delta opioid receptors. Would you say that's your favorite receptor to work on?
Yeah, so I feel like I really love working on opioid receptors in general. And most of my research is focused on mu and delta opioid receptors. But yeah, I feel like the delta opioid receptor is my baby. I've been working on it for almost 20 years under lots of different contexts. And I just really like working on it.
It's fascinating that you can work on one GPCR and work on it on different contexts at the same time. And still, there is more to learn on these receptors. What do we know about delta opioid receptors and opioid receptors in general in the context of migraine and headache?
Yeah, so, what's really interesting. So I think what we know about delta opioid receptors is that, based on my work and based on other people's work, it seems that activation of the delta opioid receptor can relieve a number of preclinical symptoms of migraine. So the pain, negative affect, aura-related symptoms. And so, I really think that it would be a really great target for the treatment of headache. And that's like a major focus of my lab, is trying to understand how the delta opioid receptor actually regulates those symptoms and looking at, what are the different proteins and peptides and receptors that are pro-migraine peptides and proteins, and then how does the delta opioid receptor regulate those things? So that, that's like been a major thing that I've been working on. In another context, I sort of look at the other half of that coin, looking at the mu opioid receptor, because there's a lot of clinical research that shows that activation of the mu opioid receptor by mu agonists exacerbates migraine, causes medication-overuse headache. And so for me, it's kind of like a yin and yang, right? Like where the mu opioid receptor seems to exacerbate headache and make it worse, whereas the delta opioid receptor seems to relieve headache and diminish its effects. So I kind of like to contrast the two just because it's sort of fun that way.
Why would one receptor do, exacerbate, and one receptor would help alleviate the pain? Are they activating different signaling pathways, or do we know anything about the reasons why these act differentially?
Yeah, that's a great question. And that's something I've really been studying recently. So, we had this nice paper looking at peptidomic, doing a peptidomic screen between animals that were treated with opioids, chronic opioids, versus animals that went through a chronic migraine model. So really trying to see what is the overlapping mechanisms on that front. And we found, for example, that there was an upregulation of a protein called PACAP, which acts on a G protein coupled receptor, PAC1, right? And so it seems that I think that mu activation sort of exacerbates, initially can be pain relieving, but as we know from, peripheral pain states from the clinic, that long-term activation of the mu opioid receptor just results in these adaptations, which then exacerbate pain and make things worse. And in migraine field, it seems one of those ways might be through this activation of PACAP. I think the delta opioid receptor is expressed in different places anatomically. It's expressed on different cell types. So although the mu and the delta are both GI protein coupled receptors, I think that where it's expressed and how it regulates these factors that increase pain or that exacerbate migraine is just different. And so in that front, on that sense, I think it just inhibits these sort of pro-migraine signals, whereas the mu opioid receptor ultimately rebounds and then results in this sort of super activation of these pro-migraine symptoms.
So in disease states, it would be more of an unbalance between the function of these receptors that creates the issues. What kind of animal models are you working on? I looked you up a little bit and I saw that you work a lot with mice and animal models. How do you get the information that you need on the delta and the mu opioid receptors from these animals?
Yeah, so most of my work now focuses on mice. I use a number of animal models of different types of headache disorders. So the one I regularly use is a model of chronic migraine. And that's a model that our lab developed where we give a known human migraine trigger to mice and then see that it causes increased pain responses in head regions, which is blocked by anti-migraine medications. So that's a model that we use commonly in our lab. But we also use other electrophysiological models, for example, of cortical spreading depression, which is a model of migraine aura, as well as things like opioid-induced hyperalgesia, and then post-traumatic headache and things like that, where basically we manipulate the animal in a way so that it reflects a little closer to what happens in the clinical condition. At the same time, we still have a general interest in pain mechanisms and emotional disorders. So, we try to use models of inflammatory pain if we're trying to contrast the sort of migraine-like effects with peripheral inflammatory mechanisms. So we'll use the CFA model, as well as looking at things like elevated plasmase, conditioned place aversion, that sort of thing.
So a lot of behavioral studies and see how these mouse models actually react to their environment and to the treatments that they have to go through, knowing that they have specific issues, pain or disease related issues. I also saw that you're interested, so you're a professor of psychiatry. I also saw that you're interested in more long-term effects of dysregulation of delta opioid receptors in behavioral. Can you tell us a little bit more about that?
Yeah, I actually think it's really important to understand the behavioral consequences of long-term use of these types of drugs, right? Because in the end, for most clinical populations, it's not often that people are just going to take a drug once and that's it. Often, they're experiencing a chronic disease or something that's, fairly frequent. They're going to end up taking the drug relatively frequently. And so I think it's really important to kind of understand, what's going to happen when people are going to be taking these drugs regularly. Are they going to end up with a rebound pain? Are they going to get tolerance? And so that's really something that I've been focusing a lot on. And I think that's where, in vivo behavioral models are really helpful because, you can kind of figure this out in a petri dish with cells from a sort of signaling mechanism, but really you don't know what happens until you put it into an animal and see what happens to them.
Do you have any evidence of these drugs, this long-term use of these drugs modifying, for example, receptor expression or any molecular changes within these animals?
Yeah, so I did a lot of work looking at earlier in my postdocs, looking at the delta opioid receptor and how whether it causes whether agonist causes internalization or not, whether it would cause tolerance, and then how the mechanism of that tolerance is regulated. And so we found, for example, that high internalizing agonists seem to just result in large downregulation of the receptor. So therefore, you'd end up with tolerance because there's no more receptor. Whereas low internalizing agonists didn't end up causing downregulation of the receptor, but did seem to result in sort of uncoupling of the receptor from, ion channels, which would then allow it to signal. So, I think that was a clear example to me of like how, depending on the agonist and what it does to the trafficking of the receptor can really change what that means physiologically.
Especially on the long term, if these animals and patients in general get these treatments over years, it's not a one-time pill fixes it all. While I was listening to you on the long-term effects, I was having a conversation earlier this week with a professor at the University of Arizona, John Streicher, and they also work on opioid receptors and in pain, in the context of pain, and we were talking about they did a study because one of his students wanted to try it out. What happens if they do intermittent fasting to their model and to see if you needed less opioids to have the same analgesic effect. And surprisingly, intermittent fasting helped decrease the amount of medication that these animals needed to help with the pain. Have you looked at anything in this context?
Well, it's interesting. Like I know Dr. Streicher and some of his work, and I think that's a really like cool thing to look at, right? Because especially a lot of people are trying intermittent fasting for weight loss, for various chronic illnesses, and I think it's really important to understand how does that affect the pharmacokinetics of drugs and generally speaking the effectiveness of drugs. And that's why I really for me, I focused a lot on trying to understand chronic disease states. Because I think that, the organism is fundamentally altered in a chronic disease state. They're not in the same state as they were if they were naive or if there was no pain or depression or whatever it is that happens to be there. And I think it's important to kind of understand what that context is because, for patients, that's what it is. They're in a chronic disease state. They didn't come to the doctor to look for drugs because they were feeling fine. So I think trying to capture all of the different physiological states that people might be experiencing is, I think, a very powerful way to develop better therapeutics. Because, I think a lot of times things fail in clinical trials, maybe because they weren't studied as carefully within these different types of physiological contexts. And I think the more we look at those chronic states or those different, physiological states, the more likely we are to get a positive hit.
Definitely. So you had mentioned that some agonists of the delta opioid receptors get the receptor internalized faster when others don't as much. And that also plays a role in the way these drugs act. What would be the ideal drug that you would be looking for in this context? I know it might be a tough question, but
Yeah. That's right. I think for the delta opioid receptor agonist, I would love something that probably didn't cause internalization. I think that internalization is, and I think there's other literature to support this, is linked to proconvulsant effects of delta agonists. So, if you can try to eliminate internalizing compounds, you eliminate that sort of downregulation of the receptor, but you can also maybe try to get rid of these adverse effects. So I think a non-internalizing delta agonist, something that had pretty good a half-life, in the maybe hours, and something that would cross the blood-brain barrier. Because I actually think that there's a lot of delta opioid receptors expressed in the periphery, in ganglia, in the spinal cord, in the trigeminal nucleus caudalis. But there's a lot of delta opioid receptors in the forebrain, and a lot in places like the amygdala, the hippocampus, the striatum, places that are really important for emotional processing. And I think the emotional processing that is important for pain processing, that, gives pain that type sort of salience. For that reason, I think it's kind of important to target those brain delta opioid receptors, not just limited to the periphery.
And how would you, again, it's a hypothetical, how would you specifically target these receptors in the brain? Since they're in different regions in the body, you really want to hit where it hurts, basically. How would you go about that?
Well, I think the nice thing with the delta agonists is that they have low abuse liability, and they also have an antidepressant anxiolytic effect. So I think the fact is that in a lot of chronic pain states and chronic migraine, there's this comorbidity with emotional disorders. So targeting brain delta opioid receptors seems like a good idea, because you can also alleviate some of these comorbidities. And in that sense, it makes me wonder if you have to be so careful about which delta opioid receptors you target. And I think that's an ongoing place of study, is trying to understand, well, which opioid receptors, where are they located that are important for targeting these different types of disease states. And that's something, our lab has been trying to do with different types of conditional knockout mice and things like that, to try to kind of get a feeling for, well, is it the ones that are in the cortex or in the, striatum, or is it just limited to the trigeminal nucleus caudalis and trigeminal ganglia? And I think I think it's interesting that all these years people have been studying opioid receptors, and we still have these fundamental questions that we still need to answer. So in that sense, I think it's kind of exciting, right? Because you can study opioid receptors for 20 years and still have a lot of holes in there that need filling.
Definitely, a lot of unanswered questions. You mentioned that with pain come emotional disorders. Can you tell us a little bit more about these comorbidity disorders that come with suffering from pain, chronic pain?
So there's no doubt, like people in chronic pain states, all sorts of different types of pain, everything from back pain to neuropathic pain, inflammatory pain, have increased comorbidities with depression and anxiety. And that you can understand that link, right? Because in the end, if you're constantly experiencing pain, that's not going to create a good mood state for you. I think in the context of migraine, that's a really strong correlation. People who suffer from chronic migraine do have this increase in anxiety and depression. I also think there's some more interesting nuances there. So, for example, a premonitory symptom before people get a migraine, one premonitory symptom is a sudden mood change. Like all of a sudden they'll go from being, fine or happy or whatever, and then like magic, they just, they get low mood, they get very cranky. It really makes you think that there's obviously a tie-in with these different mechanisms. I don't think that it's simply that, you're in pain or you've got this chronic condition, and so therefore you're going to be depressed. I think there's definitely a connection there, but I think there's also a circuit connection there, which then ends up, there's a lot of epidemiological studies that show that people who have comorbidity with emotional disorders suffer from greater pain. And there's a link there that's not just, I'm in more pain and therefore I'm more depressed. There's some other thing that sort of combines the two, which I think a lot of people are kind of exploring right now. And that's where things like the kappa opioid receptor seems to be popping up a lot.
Is there any evidence that actually the interaction, let's call it an interaction between pain and, for example, depression, that it goes both ways? I am depressed, therefore I am in pain. And do we have any idea on the molecular determinants of depression and as a consequence of, becoming someone who has chronic pain?
So, for example, there's been some really nice work recently from Jose Moron Concepcion's lab and from Kathy Cahill's lab showing that, there's an upregulation of kappa opioid receptor function and and dynorphin tone, which seems to be regulating that sort of emotional distress that's associated with pain. So it seems like there's this link with these negative feedback loops that then, are upregulated in the chronic pain state and then feed into the depression and anxiety that might be associated with them. And so I think like that's, and kappa opioid receptors, for example, have been well known to be upregulated in stressful situations, for example, or, when animals are abstinent from drugs of abuse. So, it feels like there's a link in there that kind of that's being explored right now. And I think holds a lot of promise, because there's just, I think there's a whole world there that's maybe in the last like 5 to 10 years has only started to be, delved into.
Getting back to the headache part and the role of delta opioid receptors, what is the difference between a migraine and a headache, for those who know?
So, I think what's really interesting in the headache field is, unlike in the pain field, the headache field is very strictly classified. So they have this international headache classification. It gets revisited on a regular basis. And essentially, a migraine has some very distinct phenotypes. So, it's often unilateral. It's often a pulsating, sharp pain. It lasts for a long period of time, more than 3-4 hours. It prevents you from doing other things like physical. It's exacerbated by sort of physical activities, and then it's often accompanied by things like nausea, photophobia, things like that, which is kind of different from a standard tension type headache, for example. And so in that sense, I think because the headache field is so clearly delineated that way, I think there's been a lot of success with clinical trials, because, it's easy to then, it's easier to classify patients as being, okay, so this group of patients has migraine, this group of patients has chronic migraine, this group of patients has post-traumatic headache, whatever the case might be. I think it just makes it a little bit easier to kind of like fine tune, the target population that you're looking for.
Are there at the moment any drugs that help prevent headache, or not headache, migraine specifically? And what do these target?
Yeah, so like a major success story in the field of migraine has been in the last couple of years, where CGRP blockers have really come out as being fantastic inhibitors of migraine. And, CGRP was shown to be upregulated, many, many years ago when people were experiencing a migraine, or being upregulated in the serum in people who have chronic migraine. And so now that research has sort of come to fruition, and it seems that CGRP antagonists, or antibodies that block CGRP or its receptor, are highly effective in migraine. And I think it's been a real success story in the field, because, first of all, CGRP receptor is a GSG protein coupled receptor, so that's kind of exciting. And I think it sort of goes to show, people often say, oh, well, we've already like got all of the low hanging fruit when it comes to the treatment of pathologies and G protein coupled receptors. And I think, I'm not so sure about that, because this CGRP, like it's just been approved the last couple of years. It seems to be working fantastically, and I think it just goes to show that matching the right receptor to the right pathophysiological state can really have some very promising consequences. And there are so many G protein coupled receptors out there that people don't really know that much about, and don't have like even good pharmacological tools to study. So I think, delving into those a little bit, I think there's a whole world there. And in the field of migraine, CGRP has definitely been like a major blockbuster.
Interesting. Yes. I mean, I saw a paper last month, and they cloned a new GPCR.
Yeah.
And there's all these orphans, and we don't even know what they're doing. And there's 400 GPCRs that we know of that are not non-olfactory. And, there's plenty of work to do.
I think of this, so recently I started having to study PACAP and PAC1 receptors, which are also GSG protein coupled receptors. And it shocked me because, PACAP is this ancient neuropeptide. It's evolutionarily conserved. It's not an orphan. We know what it is. Like it's it's been identified. We know what it binds to. Yet the tools to study it and what exactly PACAP and PAC1 does, still not that clear. I mean, it's only now emerging in the last 5 years or so. It's only really started to come out what it can do, and really the breadth of it, I think, is still untapped. And you just think, well, that's not even an orphan receptor. That's something that's been sitting around forever. So I think there's a lot of work that can be done, which I think makes it kind of exciting.
Yeah, which answers one of my questions that I had for you is, do you think GPCRs are still a good drug target? And I think we all agree on the fact that they are. What do you think is missing for us to be able to better target GPCRs and to use them more as drug targets in the clinic?
I think that for a long time, I think there was this idea that G protein coupled receptors were sort of simple, that they acted through their G protein and that's what they did. And now I think, obviously, there's a lot more understanding about the complexity of G protein coupled receptors in terms of, where they are in the cell, the fact that they can signal when they're intracellular, how they interact with different types of scaffolding proteins like beta-arrestins, what cell type they're on can change, what G protein they bind to. And I think that that's sort of where the complexity lies. And I think trying to really understand that complexity and how that complexity might change in chronic conditions is I think some of the challenges that really lay in that field. And I think the other thing for me personally, my bugaboo is antibodies, because in my field it's a major problem. I think every person I know who studies G protein coupled receptors will tell you, oh, the antibodies suck for the G protein coupled receptors. And I just think that it really just clouds up the picture, because on some level we're still arguing about, well, where is this G protein coupled receptor? What cell type is it on? Is it on the cell surface? Is it inside the cell? Where is it? And I mean, I think the fact that we can't say that is kind of crazy to me, and that we don't have the resolution, for many G protein coupled receptors to even tell you that fundamental thing. Forget about what it binds to as a signaling partner, or whether it signals when it goes inside the cell. I think is just such a pitfall.
Yeah, figuring out where it is and what it does and what other proteins it associates with, depending on the cellular environment. And you make a good point mentioning that if you're in a chronic disease state, it's important also to study and to differentiate the compartmentalization of the receptors, or how those cells, they're different basically than what you would get from someone or from an organism that is not under that chronic pain.
Right, and that's one of the reasons I really like the delta opioid receptor, because it seems to be upregulated in these chronic pain states, in these chronic stress situations, and it almost seems to me like it's like a defense mechanism that your body has, that it sort of increases its functional coupling or increases its receptor number when they're in these sort of threatened states. And I think that's why it's kind of a good target for certain types of diseases, because, it does get upregulated. And so therefore, if you target it in that particular chronic condition, you're more likely to have success than just targeting it under other types of conditions where it wouldn't be upregulated in.
I was talking to Dr. Graciela Pinheiro from the University of Montreal, and as you know, she studies opioid receptors, and we were having this discussion about a couple of years ago, there was this whole movement of getting biased ligands to the opioid receptors. And it turns out that it's not as black and white as you would think. What are your thoughts about biased ligands in the context of headache? Would it be useful? I'm curious.
Yeah, that's a great question. I think it's interesting to see how this whole biased ligand story has the tone of it has changed over the last several years. Where, you're right. Once it was oh, we're going to solve all our problems if we just examine the bias and then bias for, things that don't have adverse effects and bias against things that do. And now we're obviously realizing that that's not the case, or not so clear cut. I think that that's one thing that I really kind of like about the field of migraine is that, there's not a lot of basic scientists in the field. And so all of the things that I know from the opioid field, from the G protein coupled receptor field, I can bring into the migraine field, and it's almost like it's sort of a new shiny thing there because people don't talk about these things in the same way. And I do think that it's important to understand ligand bias, and I think it's important that it's clear to know what the ligand is likely to be doing if you're developing it for treatment in patients. Whether it's important or not, I don't know. And I think this is where it's really important to try to screen different ligands and understand what their qualities are, so that you can kind of at least try to target something that has fewer adverse effects. I think thinking that one signaling cascade is going to, cutting out one signaling cascade, like the beta arrestins, is going to get rid of all of those adverse effects, we're now realizing is sort of naive. But, I think it's evolving, and I don't necessarily think it needs to be thrown out conceptually just because it hasn't quite worked out the way we thought it would work out. I think we just have to understand that, physiologically, we're pretty complex.
I think it should be taken with a grain of salt. Who knows, maybe these ligands activate another pathway that we haven't been exploring, and we've been focused on beta arrestin and G protein. Maybe there was something else happening at the same time. These receptors are getting activated by these ligands. Speaking of ligands, I did ask many people I interviewed, and when in their respective fields, about what would the best ligand look like. You had mentioned that you'd want, for example, an agonist that doesn't internalize the receptor so fast. What are your thoughts about allosteric modulators in the context of altering opioid receptors in headache disorders?
Yeah, I think that allosteric modulators are really cool. And I think that it could be useful in headache disorders, especially because, there is evidence that there's upregulation of different types of endogenous opioids in headache when people are suffering from migraine, for example. And then in our own peptidomic screen, we also found there was upregulation of things like enkephalin and stuff like that. So if there'd be a way to sort of capitalize on this endogenous system by then trying to upregulate the function without activating the receptor directly through an agonist, I think that there would be a lot of promise there. I think the problem is that it's a little harder to identify allosteric modulators. And I feel like they're a little more, it's a little less stable in a sense, because, you then have to rely on there being enough of the endogenous ligand to then, increase or decrease, depending on whether you have a positive or negative allosteric modulator. And I think that, endogenous ligands are something that do change a lot in chronic disease states, and making sure that they're always, being sure that they're always going to be present in sufficient amount, in a person that's suffering from that condition is maybe the thing that makes it a little harder for the development of allosteric modulators. But I do think that there's definitely value there, and I think it's not as fully explored as just developing a straightforward agonist or antagonist.
Yeah, it's harder also to find out where these allosteric sites are to bind these receptors, and that's one of the difficulties of this. But I can very, very much envision having an allosteric modulator that's, for example, a partial agonist without the endogenous ligand, and then having the endogenous ligand or ligands, act on the receptor with the allosteric modulator and specifically do what we need them to do. Although for that, as you mentioned, you need a lot of information before we can either we can design these and also pinpoint what are the effects that we're looking for.
Right.
I looked at a couple of your papers. I want to get back a little bit to the more technical side of the research that you do in the lab. So you do have mouse models. These mice get treated with different drugs. They have chronic pain. What are the readouts that you use to look at the effects of these medications?
Yeah, so a readout that we use all of the time is mechanical allodynia. So that's basically, we use these von Frey hair filaments, which are these very thin fibers where we poke the animal periorbitally, so just sort of in their forehead region, and see whether the animal will withdraw their head or shake their head or, bat their head. So we use that quite frequently. We also try to do models that are things like conditioned place aversion, trying to get at non-evoked responses to see whether, for example, being in chronic pain increases conditioned place aversion, and then if you give a drug that can block the pain, if they have less conditioned place aversion. And I think, I think in the pain field, people have been trying to move towards having both types of models, just because, the human pain experience is an emotional experience as much as it is a nociceptive one. So we try to sort of get at that. And then the other thing is we do a lot of, we do things like cortical spreading depression, which are these waves of, that cross across the cortex that are thought to be correlate of migraine aura. And so in that sense, we're looking at visually these waves crossing the cortex in response to potassium chloride stimulation, but as well as looking at local field potential recording. So those are some of the things that we do in vivo. And then of course, ex vivo, we'll do a lot of sort of, RNA scope and immunohistochemistry and things like that, just to try to understand the mechanism of how we got the behavior.
Yeah, so just to sum up the behavior that you're looking for in these mice, you're trying to mimic as much as possible as to what happens when a human being is in pain.
Yeah, exactly. Yeah.
You had mentioned when we started the conversation that you did your PhD at McGill, then you did a postdoc at AstraZeneca, and then you traveled to France, and then back to the US, and then back. Well, actually, you stayed in the US now that I think of it. What would be your advice for young scientists or junior scientists who want to contribute in the field?
I would say, come on down. There's so many cool things to study. And the nice thing with G protein coupled receptors is you can study it on so many different levels, right? So you can look at it at the really minute molecular, micromolecular level with things like, the crystal structure. But then you can also look at it at a very macro level when you do things like clinical, trials or clinical studies, because so many drugs act at G protein coupled receptors. And then you can do everything in between, right? And I just feel like there's so many things that need to be studied, and there's just a lot of room to study it. Plus, you can basically look at anything you're interested in physiologically, right? I mean, G protein coupled receptors are involved with pretty much every physiological function. So whether you're interested in the brain or the gut or the heart, you can figure something out. I would also say the other nice thing about for younger people, getting the field is just, I think my trajectory really shows this, right? Is that it's a great way doing, science, doing your PhD, doing postdocs, of, traveling to different places, collaborating with new people, using your ideas in new spaces. So, for example, with me entering migraine, I didn't know anything about migraine when I started studying it, but it's just because I happen to have this collaboration, and then I could bring what I know about opioid receptors and G protein coupled receptors into this new space. And I think I think that people are really open, and there's a lot of opportunities if you're just willing to kind of look into them and just take them. So, I think it's a cool space to be in, and there's a lot of cool, smart people to work with.
How much do you think your openness to collaborate and to talk to people and your love for GPCRs got you to where you are today?
Oh, you know what? I think it was essential. And I honestly, I sort of try to tell my students this all of the time. And I think it is something that I learned through the different mentors that I myself had, is that, you've got to be open to sharing your tools, your science, your knowledge. And you have to be open to receiving other people's knowledge and their ideas. And sometimes they might not jive with what you think is the case. And, sometimes you might be coming at things from different perspectives, but I think being open to listening to those differences is just what makes your science better. And it's what allows you to then go on and be successful, because, you then make these connections and you have these interactions and, make great collaborations. And I just think that is something that I really think is extremely important in science. You have to be ready to collaborate. You have to be ready to work with new people and just listen to what they have to say.
Agreed. Every great collaboration I had Actually, every great conversation I had often led to, a discussion, an introduction, and it ended up in great papers and getting to know people from different fields. And as you had mentioned, you started working on GPCRs, you got interested in opioid receptors, and now you work on opioid receptors in the headache field because you met someone who was in the headache field.
Right. And, and I also have I have papers in the alcohol field, and it's just because, I'm happy to collaborate with people. So it's oh, well, why not? It's just, I know something and it might be useful to you, and you know something, it might be useful to me. So why don't we just put them all together? It doesn't take a lot of effort sometimes, and you can end up with some really cool science.
Yeah. And on the long term, it can benefit human health.
Right.
And that's the goal.
Right.
We were talking about, collaboration and you meeting this physician who worked on headache, and it kind of, the way you were talking about it, it kind of felt like it was an aha moment for you.
Yeah.
What are the other aha moments that you had during your career where you said, well, wow, this changed my view of things?
Yeah. So I would say the first aha moment I had was in my postdoc in Brigitte's lab in Strasbourg. And that was where I really was blown away at the idea that not all agonists were the same. And that even though they might end up with the same behavioral endpoint, that they got there through different ways. So, whether ligand caused signaling mechanisms that ultimately resulted in pain relief and then in the development of tolerance. So for me, that was a real aha moment to go, oh man, I really need to dig into the ligands that I use a little more carefully, because there's a lot of diversity there, and you can't just assume that because they all bind in the same pocket, they're all doing the same thing. I think the second aha moment for me was really when I started to get introduced to this migraine field and going, oh my gosh, there's not a lot of basic scientists here, and it's kind of like the Wild West. And so, you can kind of bring whatever ideas you have from whatever field you're in, and just look at and see if you can apply it to this headache lens. And especially I think in migraine, because there's so many different changes physiologically that people experience before, during, and after they have a migraine, that, you can come at it from so many different fields, not just a pain lens. And you can kind of bring these ideas here, and it's like a discovery of, riches in what you can do in it. And then I think for me, the last aha moment was really the excitement that, delta agonists, maybe this is the home for delta agonists, right? There's been failed clinical trials for delta agonists for the treatment of pain, for the treatment of depression. And I think that this makes, people wary to kind of go there. And I just feel well, but maybe what we've been looking for, maybe the disease state where delta ligands would really be effective would be in headache disorders and in migraine specifically. And maybe this is where it all comes together, right? Because there is this emotional component, there is this pain component, and where delta opioid receptors are expressed, I think really strongly suggests that this might be useful. So, yeah, I don't know. I really like these aha moments, and I just love the small ones and the big ones, and I think that's a scientist. That's kind of what we live for, right?
Definitely. And you get to learn from them, and it actually shapes your view of the science that you're doing.
Yeah.
One other question I wanted to ask you about is, how do you think we could, and it's not necessarily science-based, but it consequently it will be, how do you think we can increase diversity in the fields of GPCRs?
Oh gosh, yeah, and this is a great question. So I think for people who are already in their early mid-career as professors and scientists, as principal investigators, I think there it's really important to make sure that we put people up for awards, that we give them opportunities to write high-impact review papers, that we cite their people's work. And I think this is true, for people of color. I think it's true for women. I think there's so many different ways in which there's underrepresentation in academia in general. And I think it's really important that we try to sort of build each other up, invite each other for talks, talk about each other's work, so that it's clear that we're acknowledging that these people are there, and that we need to support them. I think at the trainee level, I think it's really important to sort of start at the level of high schools and at the level of undergraduates, where, we just go and explain to people that, look, you can get a PhD, you can get graduate school for free. Not only is it for free, somebody's going to pay you to do it. And how many different, fields can you be in where someone's literally going to pay you to get your higher education? And I think, especially in underserved communities, that is so important. And I think if we can really just, sell it from that perspective, that, look, this is an opportunity for you to really grow, and it's not going to cost you anything, Plus, I think it's important to just also really tell younger people that, just because you get a PhD in science doesn't mean that you're going to have to be an academic scientist, right? There's so many different fields you can be in, but the skills that you learn are so important. You're going to learn to give a good talk. You're going to learn to write effectively. You're going to learn to think critically. These are things that you can apply to so many different aspects of life. So, I think the more we try to sort of really get in there to these underrepresented populations and try to sell it from these aspects, I think would just be a very practical way to build people up.
You touched on two very important points on this. One is making sure that from a very young age, all the children, high school students, primary students, independently on where they come from and what their gender is, that they know that science exists, that they can get into science, and their color, their ethnicity, their financial background doesn't matter, and giving them opportunities to actually get access to this kind of education. As an undergraduate, I was involved at the University of Montreal in a project where we welcomed high school students to the lab. And let me tell you, I will always remember these two boys, and incidentally, they were boys, and they did DNA preps.
Yep.
We spent a day doing DNA preps, and they went home with their DNA with a little Eppendorf tube. But I don't know for them, but for me, it was a fantastic experience because I was able to teach them something, but I'm sure that it also gave them an idea on what waits for them if they want to go into a career in science.
Yeah, that's right. And, I think especially in underserved regions, in, Hispanic groups and Black groups, in women, in young girls, I think a lot of times people don't even realize that this is a viable career option, and that it's cool and interesting and fun, and you can do some really neat, challenging things that develop as an individual. But I think if you don't even know that possibility exists, then how would you ever think to go in that direction? And that's why I think from a young age, it's really, really critical that people start to be exposed to it.
Exactly. And the second point that I really like that you touched upon was the fact that just because you're doing a PhD or you're doing 1 or 2 postdocs doesn't mean you have to become a professor. Especially with today's environment where professorships are very rare to come upon, and you need the stellar track record, but that doesn't mean that you have to be that. There are so many career options. You can work in finance, you can work in business development, you can be a consultant. You learn so many what they call transferable skills that you can basically do anything because you get a doctorate in learning. And I think we don't emphasize it enough nowadays.
Right. And I think, you just have to look at the statistics. Like, as you said, how many PhD students actually go on to stay in academia or even in some sort of science type job? I mean, not that high a percentage. And so I think it's just important to make sure that those voices are heard, and that those, career fair type things don't just happen once someone's already in graduate school, but actually start happening when people are at their undergraduate level or even at the high school level, to go, hey, look, get that PhD, and there's a lot of things you can do with it.
Exactly. And I think getting that information is also the responsibility of the school, of the university, and helping students succeed and hopefully do a job that they love and do it the best they can because they love it.
Right. Yeah. But this is where I think, like, those people that are underrepresented, that are in academia or that are in these jobs, I think they need that support to develop themselves and keep supporting themselves, and also kind of be acknowledged for the fact that they are serving as mentors, right? And they're serving as mentors to a population that's much bigger than the standard population, right? Like, I mean, I find as a woman, I'm often asked to be on committees and things like that because, well, there's no women here, so we need a woman. Oh, and you're kind of brown, so that helps too, right? So, but that can end up being like a heavy responsibility over time, right? And it means that from an administrative perspective, it can become like a heavy load to carry. And I think that's where, departments need to be a little more thoughtful about understanding that, okay, well, if we're going to keep asking this woman or this Black person or this Hispanic person to be on all of these different committees, well, then we need to acknowledge that there's only 24 hours in the day. How can we support these people so that, that they can do those things, those important, places where they have to represent, but can also kind of get what they got into the field for.
When you have job openings in your team, where do you advertise them? And before you answer that question, I wanted to let you know, and also our audience, is that we do have a career page, drgpcr.com/career. It's free. It's for anyone who has a GPCR related job ad. It can be in industry, it can be in academia. The reason we created it is because oftentimes when you're a GPCR aficionado and you want to continue working on GPCRs, it gets very difficult sometimes to find a position that's directly related to either the receptor you love or to the field that you love.
Yeah, well, so I'm definitely going to put job ads on there then. So, yes, and then, so the other way that I tend to advertise is, so I'll do a lot of word of mouth, like I'll ask people in the field, do you know someone? Otherwise, I will advertise on Twitter, as well as on, so I go to an International Narcotics Research Conference, the INRC. There's a listserv there. So I will tend to put my job ads on the INRC. Worst case scenario, like I'll also go for neuro jobs if I'm looking more broadly. But it's, it's often these sort of niche things like a GPCR site, like the INRC, where I tend to get the best candidates because, you're right. That's where the people who are skilled in the things that I'm looking for are looking themselves.
Exactly. And I think it can be easier for everyone to find each other, because from a candidate perspective, it's hard for you to find the job that interests you, but also from an employer perspective, it is also as difficult to find the candidate that can, succeed in that position and brings the tools and the techniques that, for example, you're looking for.
Right. Absolutely.
And there is a gap, and candidates and job posters don't know that they're actually suffering from the same issues.
Yeah,
and
that's right.
that's why we created this job ad. I know we're in a COVID time, and we're not holding a lot of in-person or no in-person conferences. But if COVID disappeared from one day to another, what are the conferences that you like to go to, and where people can find you?
So, what I think is interesting is even in this COVID time, so many conferences have moved to virtual platforms. So one conference that I go to religiously is the INRC, the International Narcotics Research Conference. And that is one that has moved to a virtual platform. I think this year it's July 22nd to the 24th. And I love that conference because it's a great way to meet literally the biggest names in the field of opioids and opioid addiction, opioid pain, in this small, intimate conference of, 200-300 people. And it's just a wonderful conference to get to know people, and the science is fantastic. So I really like that conference. I tend to also like the molecular pharmacology Gordon conference. That one I think is a really good one for figuring out what's happening in the broader field of G protein coupled receptors. And then more recently, I've started going to a pain. There's like a pain 2020 monthly meeting that I've started to go to just to sort of see like what's the up and coming things that are happening in pain. What I would also encourage young people to do is that in a way we can really capitalize on the fact that everything is virtual now, because if you want to hear the science from somebody that, maybe you're a little afraid or think that they would normally not have the time to come and give your department a talk. I mean, now you're not asking that much of people's time. You're just saying, hey, can you come and give a talk for an hour through Zoom? And I think a lot of people are really open to having these virtual exchanges, which I think is fantastic.
Definitely. And that's why we're organizing, I don't know if you've seen on Twitter, but we're organizing a GPCR summit in September 14th,
Yes.
18th. We're excited about it. And since it's virtual, it's going to give access to a lot of people from all around the world, to speakers, to be able to learn what happened in the past year in the GPCR field. And it's independent on whether you're working on pain or whether you're working on ATR awareness, receptor, beta, adrenergic. Everyone is welcome to sign up. So the website is drgpcr.com/summit-2020. I believe you signed up. I'm not sure. I've been getting all these emails, but it's an exciting meeting.
Yeah. Well, I look forward to it.
Awesome. If I can add one more thing, speaking of meetings, we had mentioned the Gordon Conference, the Molecular Pharmacology. It's also a small meeting. One of my favorites, and I'm sure you know this, it's the Great Lakes GPCR meeting.
Yes.
Unfortunately, it's been postponed until next year, although we're in discussions with them to see if we can combine our collective efforts and have the summit work with the Great Lakes GPCR meeting. And I think that's also a fantastic meeting if you want to know what happens in the field.
Agreed. Completely agreed. I've had some wonderful interactions at the Great Lakes meeting. Yeah.
Fantastic.
I like these smaller meetings. I think that that's the best place for really getting into like the details of the science. And also I think for trainees, like to just make connections with people.
Exactly. It's a casual meeting where you can talk to a Nobel laureate around a beer, which you can't do often at these bigger conferences. And it's like going home, as I usually say.
Yeah.
All right. Thank you so much, Amynah. It was really wonderful having you.
Thank you for having me. I really appreciate it. I had fun.
I had fun too. Thank you so much. Take care.
Okay, take care.
Bye. Hi, Amynah. Welcome back.
Hi, how are you?
Good, thank you. It's so good to have you with us today. I wanted to reach back out and ask you how you were doing since this whole COVID situation exploded. How are you? What's going on?
Yeah, so this whole like global pandemic thing has certainly put a wrench in a lot of things. I've been so my lab was shut down for three months, and it's only been in the last few weeks that we've been able to go back to the lab in some capacity. And I think that it's been really, I think the first 3 months were actually not so bad for me because it was an opportunity for me to write grants, catch up on some papers. But I have noticed that now I'm realizing that this is an endurance test and I need to step up to that in a different way. The novelty of working at home has completely worn off. Having to have this sort of hybrid work environment where, I sometimes go in, I sometimes don't, is disruptive. So I think it's certainly been a challenge, but I'm happy that we're back in the lab, and I'm happy that especially for my students, they can sort of wrap up things, they can make the deadlines that they wanted to make in order for them to graduate or to start their PhDs. I'm sort of sad about the fact that generally speaking, like within the labs, there's just everyone's kind of on edge, right? Nobody wants to do something that could then endanger someone or is non-compliant with the new rules and regulations. And yet at the same time, we're all like desperate for some social interaction and chit-chatting with the postdoc in the next lab or whatever. And so I think it's sort of an interesting time to see how we all navigate. Once was, science is based on people collaborating and chit-chatting with one another and the small interactions that you have on a regular basis. And I think it's going to be interesting to see how we try to maintain those in a socially distant world.
Yeah, it's been difficult. I was talking to Dr. Goodkin, and he had mentioned that he doesn't like the word social distancing. They did what they called physical distancing. But it's hard because we're social animals. We like to chit-chat, and that's when you get your best idea, by going for coffee with the postdoc from next door.
Yes, totally. And I find, especially for me, like I get a lot of stimulation and like new ideas, and just by talking to my colleagues or talking to, people in my lab or other labs about the idea that I might have. And if I can't do that spontaneously or easily, I have noticed it's a little harder for me than, it used to be. And I think that's something I'm trying to figure out.
And how have you been in contact with your students? Have you had regular lab meetings, beer hours?
Yeah. So we've been maintaining weekly lab meetings. And then I try to meet with my students, sort of on a weekly or as needed basis to make sure, that they're on top of writing the intro to that paper, or, what the plan is for whatever experiments or reanalysis of data that they need to do, which I think is really important because, especially when we were all in lockdown, you've lost this major part of your life, right? So I think it's really important to try to make sure that we know we're all there for each other, even if it is just through a Zoom meeting. I think one nice thing that has come out of it all is that I started this virtual science exchange with maybe about 4 or 5 other labs in Canada and the United States that we all kind of work on opioid receptors. And so we've just started meeting on Fridays, and different trainees in our lab will give the talk, and it's been a great way for me and the rest of the labs to catch up on what people are doing and kind of hear it in a more nitty gritty detail that I wouldn't get even at a conference. This I get like literally a 45-minute talk on some project that a lab is working on. So I think it's opened up some possibilities, which is good.
And it remains a more casual environment doing it through Zoom. I've seen videos of people telling me, I was talking to my boss and then the cat came through the screen, and it breaks that ice in this sense. There is some positivity coming out of the social distancing.
There's no question. Like, I have literally had to stop, the talk that I was giving to a department and go, can you just give me a minute? I need to go yell at my children. Like, and I think that's great because, I think it just goes to show we're all human and we're all just trying to get by it in whatever way we can.
Exactly. So you had mentioned that it's been a couple of weeks when you're gradually going back to the lab. What's the new normal look like?
Yeah, so the new normal looks like that we have to be very careful how we schedule people in the lab. So, we can't have more than 3 people in the actual lab space. So people have to kind of coordinate with one another that, I'm going to be in the animal colony for most of the day, or, I'm going to be at the cryostat for most of the day. So there's a level of micromanaging, I think, that has been introduced to the whole thing that I personally didn't like in my lab. For me, I'm a little more hands-off in the sense that I like to know that people, talk about what are the experiments that are people doing, but I'm not so concerned about, I'm going to do this like from 9 to 12, Monday to Friday or something like that. And so I think now, we're all more aware of what everybody's doing and having to be more careful about the timing because of that. Then, of course, the other thing is we've had to get, everyone's wearing masks, and then we have to clean everything, 100 times a day. I had to buy webcams for all of the lab computers so that, if someone was at the lab or at home, they'd be able to like come into Zoom journal clubs or Zoom meetings. So that's all sort of new and different for us. And so, but I think we're managing.
Were you able to maintain all your mouse colonies intact during the whole lockdown?
No. So we basically, before we went into lockdown, we did, a massive cull. And it was really heartbreaking, like, to think of all of these animals that just had to euthanize because there was no way that we would be able to maintain them for so long. I managed to keep, breeders going, and we were allowed to come in during the quarantine to keep an eye on them and wean animals and stuff like that. So I think that was really good. But there's no question, like, getting my animal colony back up to speed is going to take several months. And I don't even have that many animals. Like, I think of some labs, where there's like 27 lines or something like that, and I can't even imagine how long it's going to take for people to get back up to speed.
They're not like hex cells that you can just thaw and
No.
And millions, and almost feel bad of throwing away all of those cells and still have millions and millions of cells.
Yes, exactly.
It's unfortunate when you work with these precious animals and their knockouts and their very specific strains of animals, and then you have to make this heartbreaking decision.
Yeah, and they're animals. You know what I mean? Like, you can kind of justify working with them if you feel like you're going to be using them towards experiments that might, like, advance science or therapy. But it's like heartbreaking that you've bred these animals and you literally they serve no purpose. Like, at this moment in time, they literally were just sacrificed. So
it's unfortunate. It's unfortunate. So, do you anticipate going back to the lab full time in the near future?
Well, so that's a great question. So right now I'm sort of working a hybrid model because fortunately my office is on a separate floor from the lab, so I don't count as a body in my lab space. And I can kind of be isolated in my own office by myself. And so I've been trying to go in like 2 or 3 times a week, especially when, I've got a lot of Zoom meetings and things like that where I needed to be a bit more quiet. I think the big question is going to be whether the kids go back to school in September, because I have 2 daughters. They're 8 and 10, and if they don't go back to school, my husband is working from home, but I would feel the need to be home a little bit more because I don't think it's fair that the entire burden of them during the day would just be left with him. I'd still try to go in on a sort of semi-regular basis, but I think that if they don't go back to school, that's going to mean me just being stuck in the house more.
Wow. I think that's the big question for all parents who have children of school.
Yeah,
children who have to go to daycare or to school, or it's been an adjustment, I have to tell you.
I think it's really disturbing, all of these studies that are showing that as the pandemic has gone by, that it's women that are putting in fewer grants, putting in fewer papers. I think in the end, even, I have a very supportive spouse. But even then, the bottom line is that childcare and home care tends to fall disproportionately on women. And I think that that's going to affect our careers more as this goes on and as children don't go back to school.
Yeah, hopefully they will go back to school. I'm optimistic, carefully optimistic. I'm definitely hopeful because it's not healthy for anyone. They need their routines. We need our routine, and they need to be with children of their age.
Yeah, agreed. And I think, I can see the loss of social interaction in them. And I'm sure I am also weirder now than I used to be because I don't have the same type of social interaction. And I think that we all need it. So hopefully they can go back to school in some sort of capacity.
We hope so. One positive thing out of being at home is that kids nowadays do not want to sit in front of the iPad anymore because they've had an overdose of sitting in front of these electronics.
Yeah, that's right. And I would say for me, one positive is that, I used to travel a lot for work before the pandemic. And, in a way, the pandemic is now, so now I don't travel that much for work. And so in a sense, it's nice that I've been able to spend some more time with my family. I mean, I still miss going to conferences or, giving talks, but you got to take the good with the bad, I guess.
Exactly. We take what we get.
Exactly. Yep.
Absolutely. Fantastic. Thank you so much again for coming back on.
Sure.
It was a great pleasure chatting with you, and all the best going back to the lab and getting those colonies up and running.
Thanks. And, I look forward to keeping an eye on your site and attending the meetings and, advertising on your site. I think it's a great initiative.
Thank you so much. We're excited to get any job ads on the website so that we can help people get their perfect match.
Yeah, agreed. Well, I'll be there.
All right. Fantastic. I look forward to it. Thank you so much, Amynah.
Yeah, thanks. Take care.
Thanks. Bye.
Thank you for listening to this Dr. GPCR podcast episode. We hope you enjoyed it. Next time on the podcast, we chat with Dr. Brian Roth from UNC Chapel Hill. If you'd like to sponsor us, visit drgpcr.com/sponsor. We truly appreciate your support, which allows us to bring you podcast episodes, newsletters, and so much more. Thank you to Dr. Amynah Pradhan, Attila Forrest, Jin Chong, Shivani Sachdev. Music by Rosa Bershish. I'm your host, Dr. Amynah Bershish. Thank you for the privilege of your time, and until next time, stay safe.